here's the second part of the missed lecture. So we'll add up to a total of the same time, length of class time we missed. So I'm sorry again, I missed class. I said I thought I might have food poisoning. I don't know. Maybe I have the flu. But here we go. And I'll just record this, have a timer going. And so again, our exam is going to be, our first exam is going to be on October 17th. And this is what to do this week in class, and the materials there. And so this is, we're going to start talking about the handout that I physically gave out in class, and I'll post it on the announcement with this recording as well. And it's a key piece of our course. It's going to introduce a lot more things, but it's built on everything really that we've been talking about. And what it's about is some of the molecular factors that set up the early frog development. And so to get there, before we start reading through it, and so the handout looks like this. It's a series of statements of molecular factor steps, and then it has a figure in it that's from a different textbook about the activation of a couple of target genes that we're going to talk about. And so what we're going to be talking about is this Xenopus again, frog, and we'll start with fertilization, and we're going to take it to this stage here. Okay, so it's going to be molecular factors that are set up and then take us to this organization here. And what we want to do is get a feeling for what is it that we're heading to. And to do that, I'm going to show you some figures from chapter 4. This one is back to chapter 1, and I talked about it in the first part of this lecture. So this first experiment is called a fate map, and we use it to see that even long before, in embryogenesis, long before cells have achieved their fate, could be honeycomb, could be brick, that means could be pancreas, could be liver, where they reach that fate, if we tag the cells and just let the embryo develop normally, we can see that these cells already have a fate, and that's indicated by this kind of color pattern, right? The cells look the same, they're the same cross-line shapes and stuff. They have different fates, and it's based on their position in that early embryo. So that's the first one, is an experiment, experimental embryology, and that experiment is called the fate. Then the next is that if we start moving these tissues around, we see some different effects on their ability to achieve their fate. The first one to show is that really early, after a cell achieves its fate, or maybe about the same time it achieves its fate, it becomes specified. And specified means that if removed from the embryo and allowed to develop, the cells will reach their fate. So before they're specified, they're unspecified. And what this experiment is showing that is that the cells, and this is very common, the cells already have a fate, but they aren't yet aware of their fate, and they need to receive communication from some other cells. So some kinds of signaling molecules need to be used to stimulate this, and this would be a change in gene expression where the cell doesn't know what it's supposed to become and then does become that. Okay, so then the next thing to say is after a cell has been specified where it can develop normally to its normal fate if just isolated in a dish, after that the cell is still undetermined, and that means that if we move the cell to a different location in the embryo, where there'll be some different signaling molecules, that cell, the fate could change. So when it's undetermined, it might be specified, leave it alone, it knows how to reach its fate, but it can still be altered to the fate of a new neighborhood. And then at a certain point it becomes determined, and that is even if you move it, it still achieves its normal fate, even if it's hearing conflicting signals and in the new environment in the embryo. So here again, changing gene expression, and all this is happening before differentiation. Could be a long time before it. So we talked about differentiation, sort of the achieving fate. That's one definition you could use for differentiation. You could say when a cell achieves its fate, terminal differentiation. And so that's this pathway, and it's kind of a classic pathway in development. Okay, so if we use these terms, we can look at this. So remember, we're going to be looking at the development of this type of body plan, where we're going to have a triploblast body plan of endoderm, mesoderm, and ectoderm. All right. So in our book, this shows an early blastula. So just to go back here. So an early blastula. So this is going to be a blastula here. So an early blastula would be here. And we can make a fate map. And it gives us a very simple result that looks like this. So this is, just to show you, this is where the blastopore will form. The blastopore doesn't form, the dent doesn't form until the start of gastrulation, but it will form here, just to give you some orientation. So that means the dorsal lip of that blastopore will be here, to give you some orientation. Okay, so this is a fate map, and the yellow is endoderm, the blue is ectoderm, and the red is mesoderm. And so what it shows us is looks like a kind of like a globe, where this would be like the northern hemisphere, and the animal pole is up here, and we can see that the ectoderm is up here, the endoderm is down here toward the south pole, and then there's a belt that I've talked before about in class, a belt around the sphere like the equator. And just to make sure you can see this clearly, this is going to be a belt, and now we're going to talk about it a bit like a belt, where right here, and nobody uses this term, it's just an informal analogy, but that's like the belt buckle. So it's a mesoderm belt around the equator, and then there's a special place, and that special place is going to become, is fated to become that dorsal lip of the blastopore. Right there, okay? So I'll use that, refer to that, okay? So this is where things are eventually going to fold in. The endoderm and the mesoderm will fold in here, and during the early part of gastrulation. But this is early blastula, and it's a fate map. The cells already have some awareness of what they're going to be. Because if you just leave the embryo alone, this is what they'll be. All right? So now I used Photoshop to make an additional figure. I'm going to show you that. So your book doesn't have this. This is a specification map. So this is where if you took these cells away from the embryo, they would reach their fate. Okay? And this is the fate they would reach. Now, let's just start with this. This is the blue ectoderm. And if we take the cells away, everything turns into epidermis. These cells right here, they're fated to form the central nervous system, CNS, the brain and spinal cord. But if we take them out of the embryo early, they don't form the nervous system. Why not? That's because they are unspecified. And we know why not. We know, we've learned in our course, that to make the nervous system, the cells have to be near the nodal cord. So what this is saying is all these cells are ectodermal cells, and they're all ready to make epidermis, to make skin. And they're unspecified. You can have them change their mind, and that's what normally happens when these cells these ectodermal cells get exposed to the nodal cord. So the nodal cord, remember, is that central finger of mesoderm that's going to be under the cells, and it's going to cause the cells to fold up into a neural tube and form the nervous system. So that's a great example where this helps you appreciate what this map does. Now here's the next thing we want to show, and that is, you can see here, see this belt, these cells are fated to form the mesoderm, but look here, the cells that are fated to form a mesoderm, if you take them out here, they'll actually form ectoderm. There's no cells yet specified to form mesoderm. And that's going to be the belt with the belt buckle. So these cells are unspecified. And then look, when you go from the early blastula to the late blastula, there's a change. These cells now became specified. What happened? These cells must have received some signals, some signaling factors that were necessary. The cells needed to be in the normal embryo environment during this period in the blastula to become specified to make the mesoderm and the notochord and somites. So this special region, right? So not just mesoderm, but some different types of mesoderm here. So let's just scroll back here and say that in the embryo, we've talked so far about this mitotic cleavage is just the cells quickly getting divided up. We've also talked about how they get divided up and they become different. So we haven't talked about this for a while, but mom initially loaded into the egg factors in different locations. So the cytoplasm is not evenly, smoothly proportioned with different factors. Some are here, some are there. And we see evidence for this now in that map, because we see that in the early blastula stage, something that mom must have provided already tells these cells in the animal pole, you should be ectoderm, and these should be endoderm. And but then during the early blastula and into that late blastula, something's happening to cause a change here. And that's the focus of the handout. And what we're going to find is that there are going to be some factors that are down here that are going to play a role in causing this change to go from here, where there is no mesoderm, to here, where a belt of mesoderm gets specified. So the handout really focuses on factors that are coming up here and that change the cell's fate here, right? Because we're changing their, well, we're not changing their fate, we are helping them to prepare for their fate, okay? Like that, all right? And so let's start looking at this handout. And it'll take us a while to walk through this, but I've written the handout as directly as I can, just like directly, because you'll end up having to memorize this, because I could ask a 20-point question on the exam that says explain the molecular steps in the early patterning of a frog embryo. And this isn't all of them, but it just gives us a sort of a minimal framework understanding of how these things work. Okay, so it starts out and it says disheveled protein is a maternal factor localized to the vaginal pole. All right, so now we have to take that first sentence and explain it. So here's an egg, and this story is different than us, right? Because we don't have big eggs like this. We'll see that there are orthologs of human proteins that play roles here. But remember that these developmental regulators are like wrenches and hammers and screwdrivers. They can do their job multiple times. And so humans, we don't have the same early development, but some of the same types of proteins that we have are used here. All right, so here it is, and it says there's a protein localized down here. And this is all one cell, so it's localized in the cytoplasm near this vaginal pole. And it's called disheveled, and it's a protein. And it's said to be a maternal factor. That's a term we haven't talked about yet in our course, so let's get a definition of that. A maternal factor is an early development regulator that cannot be rescued by the paternal genome. Okay, so what's this saying? To start out saying it, we've already talked about this idea a few times. So we've said that this is a product that mom made, right? It doesn't have anything to do with the egg yet because it's a mature egg prior to fertilization. And so this disheveled protein had to be made in mom during egg maturation. Everything in the egg was made in mom. Any of the proteins we talk about that are in here were made by mom. And remember we said mom packed in a lot of nutrients, a lot of nucleotides to synthesize a lot of DNA, a lot of amino acids to synthesize a lot of proteins, and also included some proteins that are developmental regulators, and disheveled's one. This was made in mom, and so mom has a disheveled gene like that. And let's see, there's the 2 chromosomes, and she got one from her mom and one from her dad, but that's going to be, the previous generation. So mom looks like this. Okay, mom needs to have a good version of the disheveled gene to put it in here. If mom has a mutation in her gene, then this is defective. Now, we're talking about the embryo, and this is the dad for that embryo, but fertilization hasn't happened. So here comes dad with disheveled gene, and let's say mom has a hypomorph, loss of function allele, and dad's got wild type disheveled. Well, this disheveled is made in mom and needs to function during embryogenesis before dad's genes are used. The gene expression doesn't happen in the embryo until that late blastula. So let's just go back to this image and have this added to this drawing. So this is when zygotic gene expression begins, and people always say it begins in earnest. Maybe there's a little bit of expression earlier than this, but here's dad's DNA arriving, and it's there, but neither mom's nor dad's DNA is getting transcribed into genes, transcribed into RNA here. The cells are just quickly dividing, and no gene expression is occurring. The proteins must have been made in mom. And even though dad's chromosomes are here, they can't help because there's no new gene expression for a while. So everything, all the proteins that get used early are called maternal factors. And the official definition is they can't be rescued by the paternal genome. That just means that's a genetic term, right? Because we're kind of doing a complementation cross between mom and dad, and the early maternal factor needs to be functional in mom. And if mom's mutant and defective for it, doesn't matter if dad's wild type, because this needs to be used and made in mom during egg maturation. So that's a really important concept. There are maternal factors in every kind of animal, except there don't seem to be no maternal factors in mammals. So remember, mammals have little tiny eggs, and our relatives, non-mammalian relatives, have big eggs, and the big eggs have lots of nutrients and lots of these maternal factors. All right, so that's the first thing, is that disheveled protein is maternal factor. It must be made in mom during egg maturation, and it's localized to the vaginal pole. It's in the cytoplasm, but not smoothly everywhere. So one thing we could do is we could draw this, which is we could just say, because one year I did this on an exam, I said, here's a mature egg before fertilization. And I always draw it like this, right? Where there comes the sperm. And then I say, on the exam, I say, it just looks like this. There's a circle for the egg and the sperm, and I say, draw the disheveled protein, and you would draw it down here. That's where it is, okay? Before fertilization, and it's a maternal factor. All right, so now, the next sentence. Cortical rotation moves some disheveled protein into the future dorsal region, the area of the dorsal lip of the blastopore. Then there's some more terms here. Okay, they're all synonymous. They're just they are occurring at a somewhat different developmental stage. So now let's talk about that. And our book talks about all this. So at fertilization, what happens if we draw this bigger is here comes the sperm fertilizes the egg. There's the sperm nucleus. Before they fuse, the two and dad's nuclei are called pronuclei. Egg pronucleus, and the egg pronucleus. So there they are. We've had fertilization, and they haven't yet fused. And so adds that. That also adds additional organelles that are going to affect the organization of the cytoplasm in the egg. And these are called centrioles. I'm just going to draw a little example of a centriole in another cell. Centrioles play an important role in mitosis. Anytime we have mitosis, so this is going to be just mitosis anytime in a eukaryote. There are the chromosomes. Let's draw these chromosomes. Here are the chromosomes. And you know that the chromosomes are going to get pulled apart during mitosis. And you probably know that the chromosomes are going to get pulled apart because there are some connections like threads, and these are microtubules, part of the cytoskeleton. These microtubules are like train tracks. They're polymers of the protein tubulin, and this entire thing is organized by these centrioles, and collectively it's called the mitotic spindle. And that's just a side note. We're not going to be tested on that, but that's just something that you should have learned in intro bio, so I'm just trying to help you make that connection. And these centrioles are microtubule organizing centers. MTOC, microtubule organizing centers is the abbreviation they go. And so that happens every time with mitosis, okay? So there are microtubules in the egg, and they're just kind of scattered around. And upon fertilization, what happens is these sperm centrioles actually reorganize the microtubules in the egg, in the zygote, single cell. So we have a lot of microtubules with this kind of like rearrangement like that. And what happens is that about 1 hour, 60 to 90 minutes after fertilization, there is a cellular process called cortical rotation. And scientists have been able to see evidence for cortical rotation for 100 years or more, because this area of the single-celled zygote, this area of the cytoplasm, changes kind of appearance, and this area is referred to as the gray crescent. Okay, and the gray crescent is kind of opposite from the site of sperm entry. So here's the sperm fertilizing the egg, because the range of this, the gray crescent is that way, and it's because cortical rotation involves a movement of some of the cytoplasm in this direction. And it's moving because of the organization of the train tracks. The train tracks are oriented, and so this material gets pulled, gets transported along these microtubules. So it's visible here. And so scientists for a long time could see, oh, something's happening over here, opposite the site of sperm entry. And just to sketch this in, this is going to eventually look like this, where that's the belt, and this region is going to become this special belt buckle, which is going to be the dorsal lip of the blastopore, which is going to be releasing factors. So our handout is heading toward explaining why this area is going to be special. Now finally, before I erase this and draw some more, remember that mitotic cleavage is going to be happening, and it's going to divide this into the hollow ball cells of the blastula. So we're going to have lots and lots of cell division, and so what we're saying is that early there's no cell, no separation of cells. This is one cell, and the cytoplasm starts out special here because this is disheveled down here, and one of the factors that moves during cortical rotation is disheveled gets moved here, and it gets activated and developed. That's going to be a big factor in our story of the molecular mechanisms to create the specified belt with belt buckle that will become the dorsal lip of the blastopore, the Spamen center, the inducer that can induce cell. These cells could, if transplanted, could induce a second embryo on the tummy of a host. All right, so then when it starts out, initially, there is another protein down here that is an inhibitor of disheveled. And it appears that the main function of cortical rotation is to move the disheveled away from the inhibitor. So the disheveled gets transported, but the inhibitor does not. And as a result, we move it away from that inhibitory region of the cytoplasm to this place where it can be active. So that's about an hour, hour and a half after fertilization. Cortical rotation creates that visible gray crescent area opposite the site of sperm entry, and that's where this disheveled has been moved by transport along the microtubules and is now active. All right, great. So disheveled, when it's active, binds to a protein called GSK3 and blocks it. So let's just go here and erase this. Remember I said, we could have a question on the exam where it's like, here's an egg, here's the sperm. So it's a mature egg, and draw disheveled. And disheveled is down here. And then the next thing is to say, what about this GSK3 protein? Where's that? Probably make it a different color. GSK3 in the mature egg is everywhere. GSK3 is a maternal factor. All these early proteins are going to be maternal factors because there's no real gene expression happening and dad's genome is not being used. So GSK3 is going to do something everywhere except for here because disheveled will block it. the shovel blocks GSK3. Oh, and so let me just, that's my little timer saying we've done enough class time. But let me just finish what happens. And blocks GSK3 from degrading a transcription factor that's a very important one we'll learn about, beta-catenin. Beta-catenin is a transcription factor, so that means it's going to have a job similar to MyoD and myogenin. It's going to activate gene expression. It's going to activate important gene expression. When genes start to be expressed, there'll be new gene expression here in this region. And this is going to become the genes that need to be expressed to make that special belt buckle, the dorsal lip of the velociform. So we're doing great. Beta-catenin comes up in our homework in our Wnt signaling pathway, as does GSK3. So this is very specific and brief here, as brief as I can make it, but this is explained more in more detail, both in the textbook and in our Wnt signaling homework that's due next Thursday. Okay, so we got to stop it here, and we'll take over on this handout on Tuesday. We'll go through this and we'll start talking about some of the features of Chapter 5, because this is really the centerpiece of Chapter 4 as best as I can tell it. Great, thanks.